for the opportunity to speak and for bringing together this really wonderful group of people. In the next 30 minutes, I'm going to do something that you're never, ever supposed to do. And I do this, I'm going to tell you a chronological story. And I do this for many reasons, one of which is that in the absence of data, I have anointed myself the motivational speaker for this meeting. So I'm actually going to tell you a story about the scientific process using the seemingly interminable search for the fine-scale distribution of crossovers in melanogaster as a backdrop. So this story starts in 2007, when I was a postdoc at Cornell University with Chip Quadro and Andy Clark. In 2007, of course, George W. Bush was president. Nancy Pelosi became the first female Speaker of the House. You'll remember that Marion Jones had to surrender 5 Olympic medals after she admitted to doping. Other notable events include the discovery of a 2,100-year-old melon, fully preserved in Japan. The last installment of the Harry Potter series was released. And for my fellow horror movie buffs, you'll recall that 2007 marked the release of the 4th installment of the Saw movie series. Now, the most notable thing for me in terms of academics in 2007 was the publication of this beautiful paper by Elizabeth Cerulli, Rich Kleinman, and Mohammed Noor in the Journal of Molecular Evolution. And in this paper, as the title suggests, they looked at fine-scale distributions of crossovers in Drosophila pseudoobscura. And I read this paper and was totally blown away by the elegant simplicity of this experiment. And I decided that I was going to start thinking more about meiotic recombination. So of course, meiotic recombination really refers to the frequency of genetic exchange between homologous chromosomes during meiosis. And as Jeff just told you, there are 2 outcomes, possible outcomes of the meiotic recombination pathway. There are crossovers and there are non-crossovers. I'm going to be talking about crossovers only. I'll use the term recombination rate, but I want you to remember that I'm not talking about non-crosser events at all. We've known for quite some time that recombination rate is a really important population genetic parameter that has a number of effects on genome evolution, including things like the efficacy of natural selection and levels of extant nucleotide diversity. And rates of meiotic recombination are variable in terms of a core scale variation on the order of chromosomes or megabases. We know that there's interspecific differences in recombination rate. We also know that there's population level variation in recombination rate at a core scale. And then of course within the genome, there's differences in the distribution of crossovers as well. With respect to fine-scale variation in recombination rate, now we're not talking about megabases, we're talking about tens of kb or hundreds of kb. There's been a huge amount of attention recently in literature on recombination hotspots. And I'm going to define recombination hotspots by these 2 features. One is that they reflect local increases in recombination rate on the order of 10 to several thousand-fold relative to background or relative to the surrounding sequence. And the other feature that I think defines hotspots is that the majority of recombination events takes place in a very small fraction of the sequence. So as Adam showed earlier, in the human genome, it's estimated that up to 80% of crossing over takes place in only 20% of the sequence. With respect to recombination rate in Drosophila, we've known for quite some time that there are no hotspots, at least in the way that I've just defined them. And that's because patterns of linkage disequilibrium in Drosophila are inconsistent with a hotspot model. With hotspots, you expect to see very long haplotype blocks that are punctuated by hotspots, and in Drosophila, linkage disequilibrium decays incredibly rapidly, on the order of a few hundred base pairs. Most approaches for estimating recombination rate in Drosophila have been statistical in nature and rely on the relationship between the physical and the genetic maps. And using these statistical approaches, many people have found core scale heterogeneity in recombination rate within the melanogaster genome, but really we're limited by the resolution of the genetic map. We only have a few hundred genetic markers, which means that there's a lot of uncertainty that remains regarding the scale at which recombination rate varies within the genome. With respect to 5-scale structure, in 2007, we knew almost nothing. There's some beautiful classic work by Chobnik and others that applied techniques like half-tetrad analysis. These techniques are very difficult to apply to any arbitrary locus in the genome, so we knew very little about the fine-scale structure of recombination rate variation in melanogaster. And then, as I mentioned, in 2007, there was this publication from the Cerulli et al. Paper where they looked at a 2 megabase region on the pseudoobscura X, and they found 40-fold variation in crossover rate in Drosophila pseudoobscura at this fine scale. So we know that there are core scale differences between pseudoobscura and melanogaster, and I really wanted to ask whether those differences can be recapitulated at a fine scale. And so I essentially took that Cerulli paper into Andy's office and I said, I think we should do this in melanogaster. And in a moment that he has probably come to regret, half a dozen times over in the last 5 years, he agreed that we should go ahead. And so we used a similar approach to the Cerulli et al. Study to create an experimental map of a particular region of the melanogaster X chromosome, and we wanted to get a map of very high resolution, and so our goal was to survey 10,000 recombination events and localize them with kilobase level precision. So the first step in this experimental approach was that we created a doubly marked strain of Drosophila melanogaster. This strain is marked with 2 visible X-linked markers that are recessive, corresponding to mutations in the genes white and echinus. White and echinus are about 1.2 megabases apart on the physical map and 4 centimorgans apart on the genetic map. And we used a 2-step crossing scheme to identify males that had a single crossover event between the genes white and echinus. So in this first cross, we take our double mutant females, we cross them to the wild type males. These F1 females are doubly heterozygous, and Drosophila recombination only happens in females, and it's these recombination events that we're going to be scoring. We take the F1 doubly heterozygous females, we back cross them to the double mutant parent. These are the possible progeny that you can recover from this cross. And the nice thing is that because males are hemizygous for the X chromosome, you can actually visually identify males that have a single crossover between white and echinus because they contain one visible marker but not the other. So they're either mutant at white and wild type at echinus, or wild type at white and mute at echinus. So we calculated the number of crosses that we need to do to get 10,000 of these things here in red. We did all the crosses. In the end, we scored about 190,000 back cross 1 progeny, and we scored all of the males for the recombinant phenotype. And unlike the 10,000 recombinants that we were expecting, we ended up with 536 recombinant males. And there are a couple reasons for this. One reason is that the observed genetic distance between white and echinus and our cross was only 0.6 centimorgans, and the published map distance is 4. So we saw a huge reduction in the rate of crossing over in our lab condition. Another reason that we got so many fewer recombinants than we expected is that it turns out the echinus phenotype is almost impossible to see, and so it requires a great deal of time to handle the fly to get them into the exact right position to the phenotype, and so we ended up throwing away half of our flies without even scoring them because there simply wasn't enough time in the universe. But 536 is better than 0, and so we decided we were going to move forward and try to create a fine-scale map of the white echinus region. And so we genotyped these 536 males at 8 single nucleotide polymorphism markers. These are markers that distinguish our 2 parental lines. And then we used MapMaker, which is just a freely available software package that takes genotype data to infer a recombination map. And these are the results from that study. We have our intervals on the x-axis that are bounded by our visible markers and also containing our 8 single nucleotide polymorphism markers. And here's recombination rate in centimorgans per megabase pair. And there are a couple things to notice about this plot. First is that the distribution of crossover events at a fine scale in this region of the melanogaster X chromosome is significantly different from uniform. However, unlike what we're seeing in Drosophila pseudoobscura, which showed 40-fold variation in crossover distribution at a fine scale, the magnitude of the variation in crossover distribution in this region on the melanogaster X is actually quite small, on the order of 3.5-fold. And this was a really unsatisfying result for us because we didn't know whether this reflected a biological difference between melanogaster and pseudoobscura, or whether it actually reflected the fact that we only had 536 males that we were sampling at only 8 single nucleotide polymorphism markers. Now, all of this came to a head in about 2009. In 2009, of course, Barack Obama took office as the President of the United States. 2009 also marked the H1N1 swine flu pandemic, Miracle on the Hudson, the death of the King of Pop, and of course the release of Saw 6. Now, we decided that we were going to do this study again, learning lessons from the not so successful first chapter of this installment. We decided we were going to do it again. We were going to pick different visible markers. And so we decided we were going to scale this up. We were going to go for 10,000 recombinants again using a different pair of markers. And the markers that we ended up selecting are, again, recessive and X-linked. And they correspond to mutations in the genes garnet and scalloped. They're a little bit farther apart on the genetic map, so we can get more recombinants per cross, but they're also a little farther apart on the physical map. So now we're talking 2.1 megabases. We use the same 2-step crossing scheme to generate recombinant males having a single crossover event between garnet and scalloped. And in the end, we identified about 6,700 recombinant males, and we were very happy. We're closer to 10,000 now, and we were also very happy because the expected map distance from the Big Red Book is 7 centimorgans, and we observed a genetic distance that was actually very similar to that. So now we have our recombinant males, and we want to identify the recombination breakpoints by genotyping these males at SNPs that differentiate our two parental lines. And now we have this problem because we don't know how to genotype 6,700 things at 10,000 markers. And so we thought about this for a very long time and simulated this a dozen different ways. And in the end, we decided that we were going to genotype these males by resequencing. So we want to target the 2.1 megabase region and get resequencing data so we can capture all of the markers in all of these recombinant males. And we had to make we had to use a couple of different techniques and tricks to kind of make this economically feasible. And the first is that we used a chip capture step. So we want to focus our sequencing efforts on the region specifically between garnet and scallop, because sequencing elsewhere in the genome doesn't help us at all. So we used the Nimblegen platform for this part of the project. It's a custom Nimblegen array. It has 385,000 spots, and each one of the oligos on that array maps to the region between garnet and scalloped. Of course, we used next generation sequencing. We were using multiplex next generation sequencing, and we were using the Illumina platform, and at the time we were using 86 base pair single end reads. Now the last kind of innovation we had was to pool individuals. So rather than sequencing individual males, we were going to group them into groups of 100, and the idea here is that you can use the read counts within a pool at any particular SNP to infer the allele frequency of that SNP. Of that allele, and then you can track the allele frequency changes across your interval, and that will tell you where the crossovers are actually happening. So to kind of walk you through this, this inference, so we have two phenotype classes of pools. I'm going to give you an example where they're wild type at the 5' end and double mutant at the 3' end. So we have a pool of 100 flies. Here are SNPs, and here on the y-axis is the frequency of the double mutant allele. So we know, because they're visibly wild type, the frequency of this double mutant allele at the 5' end of this locus should be 0. If we go to the first SNP and we see that the frequency of the double mutant allele has increased to 5%, we can infer that 5% of the crossover events localize to the region between SNP 0 and SNP 1. If we go to SNP 2 and the frequency of the double mutant allele is 20%, we can infer that 15% of the recombination events happen between SNP 1 and SNP 2. Oh, my numbers are missing. We can march our way across this entire interval, and using the change in allele frequency from adjacent SNPs, we can actually translate this into a fine scale recombination map with respect to centimorgans per megabase pair. So this is the overview of this project. We ended up with 6,600 males that we could group into 66 pools of 100. We constructed barcoded libraries from all 66 of these pools. We grouped 11 different libraries together to make 6 super pools. We hybridized each super pool to our Nimblegen array to enrich our genomic DNA library for the garnet scalloped region. We Illumina sequenced the enriched product, and then we used a suite of bioinformatic programs to estimate allele frequency based on read counts and then translate that to a recombination map. Now, we ran into a little bit of trouble with these data, and I'll explain why. So this is the raw data from one of those 66 pools. Okay? We have physical position on the x-axis, and we have the double mutant allele frequency on the y-axis. Each one of these red dots is a SNP. Okay? Now this pool of flies is wild type at the 5' end and double mutant at the 3' end. And you can see that most of the points on this plot do follow this kind of monotonically increasing line. But you also see that there's a huge amount of variance in the estimated allele frequency from our read count data. And it's way greater than binomial. We were assuming that we were going to get binomial allele frequency, variance in allele frequency, but this is far greater than binomial, which makes it very difficult to model. Now, one of the reasons that this variance in allele frequency is so great is that we introduced a bias when we used that Nimblegen array. So the Nimblegen array is, of course, based on the reference. We have 2 different parental lines. One of them actually is African in origin, which means that they're going to have different hybridization efficiencies to our array, which means that you don't get an accurate assessment of allele frequency when you're just basing it on read counts. Now, we knew that there would be a hybridization bias between our two lines. We actually thought it was going to be systematic and that we could statistically correct for it, but it's actually not systematic at all. It's completely stochastic, and so it's actually very difficult to turn these data into a recombination map. So at this point, this was 2010, and I had thought that this was the most catastrophic failure of an experiment in the history of the world. 2 years later, it turns out there are usable data. There is a usable signal in those data, and we've teamed up with a statistical genius, Eric Stone, to help extract that signal, and we now have a signal that we believe. But in 2010, when I made that plot, I thought this was over. I was done. So 2010 was a coming of age year for me. I had my first child. We bought a car to accommodate said child. My husband and I bought a house because we both started faculty positions at North Carolina State University. And of course, to complete the yuppie transformation, we both bought iPhones. I should say, lest you think that the rest of the world came to a grinding halt, you'll be pleased to know that the final chapter, SAW 3D, was released in 2010. So in 2010, we start my lab at North Carolina State, and I decided that I was going to do this experiment again, learning from what went wrong the second time and the first time, because I was sure that the third time was going to do the trick. So we used the same loci, we used the same crossing design. The only different thing that we did this time was that rather than using a single wild type line for this male, we actually are using males from 16 different lines. There are 8 from a Raleigh population and 8 from an ancestral population in Uganda. So we do this set of crosses 16 times to generate recombinant males. And we screened about 130,000 males, and we identified just shy of 10,000 recombinants finally. And we got about 500 recombinant males for each one of those 16 lines in that first cross. The recombinational distance between garnet and scalloped in each one of these 16 crosses is really hovering around the published distance of 7 centimorgans. So now we have to genotype these, and we know we can't pool them, so we have to resequence individuals. And so we decided that we were going to team up with Peter Endolfato, who's at Princeton University, and take advantage of this approach that he has called multiplex shotgun genotyping. And he calls it that because it really is a combination of shotgun sequencing and rad tag sequencing. So you use an enzymatic digestion using a common cutter to shear your DNA, and then you basically shotgun sequence the rest of your reads, the rest of the genome that you're enriched for sites that are near MSC1 sites. So we did a barcoded individual genomic library prep followed by this multiplex shotgun genotyping approach, and we used 100 base pair single end reads. Now again, we want to focus our reads on the region between garnet and scalloped, and so we again want to enrich our genomic DNA library for this region, and so we turned to the Agilent platform, and there are a lot of advantages of solution phase over fixed array that I'm happy to discuss later, but we use this Agilent solution phase capture, and when the Agilent rep came to my office to talk about what we wanted to do, I told him, I said, we want to take 384 flies and we want to put them in your solution phase capture, and we want to pull out DNA from all 384 flies. He told me I was crazy. He's like, you can't do that. It's like, well, why not? He said, well, the molarity of the baits isn't high enough. I said, well, what's the molarity of the bait? I said, that's proprietary. Okay, so he said, maybe you could get away with multiplexing 10, but anything more than that, I was wasting my time and wasting my money. And so I decided that he hadn't convinced me that it wasn't going to work and that I was going to try it anyway. And Peter was on board. So Peter was actually very excited about this project because they'd never applied MSG to a small region of the genome. They'd only done it on whole genome samples. And they'd also never multiplexed up to 384. They'd only gone as high as 96. And so he thought it would work for 384, but he wasn't actually sure. So we decided we were going to do a pilot. Let's take 384 of these recombinant males, do the MSG, do the Agilent enrichment, and see what happens, okay? So this is what happened. Okay, so this is the number of reads on the x-axis. This is coming off of a single HiSeq, a lane of HiSeq, and the number of libraries on the y-axis. And so we have 384 libraries, and only 17 of them did we get representation below 10,000 reads. 10,000 reads is still 0.5x coverage of the garnet scalloped region. And most of our libraries are giving us on the order of 10x coverage of the garnet scallop region. So this Agilent platform actually did work at pulling out DNA from every single fly in that pool of 384, which is actually really amazing to me. So now we have these reads, they map to the right thing, and we want to figure out where the recombination breakpoints happen. So we take each read, we map it to the 2 known parental sequences, and then we basically use a hidden Markov model to identify the parent of origin for each single nucleotide polymorphism in our sample. So in a perfect world, these are idealized data, right? This is what it would look like if things were perfect. This is the physical position here, and you go along with your hidden Markov model, and it says that it's mapping to parent 1, parent 1, parent 1, parent 1, and then all of a sudden, everything switches and everything maps to parent 2. You would infer that your recombination breakpoint is right here at this transition between the red and the blue. Okay? So we took this approach and we applied it to those 384 libraries, and this is just representative data. These are 4, I'm not kidding, randomly selected individuals from that pilot. And what you can see, these 2 are the same phenotype class. They start out, all the reads map to parent 1, parent 1, parent 1, and they swap over and they start marrying parent 2. These are the reciprocal phenotype class. They all start mapping out to the wild type parent, and then the reads map to the double mutant parent. So depending on where the SNPs are, the SNP density, you can actually localize the recombination breakpoint within some window, and that window depends on inner marker distance. This is a plot of the kind of basic our confidence in where those breakpoints are resolving to in base pairs, and you can see the distribution here. This blue line is the median, and you can see for more than half of our libraries we can resolve that breakpoint with a precision of less than 5 kb. The mean here in black is a little bit higher. That's really because of these are the 17 libraries that we have pretty low coverage, and so the window of resolution is actually quite large. So we're actually incredibly happy with how this turned out, and I think that that this approach is going to be really effective for making a fine-scale map of this region of the melanogaster X chromosome. So the rest of the story has yet to be written. We need to genotype the remaining 9,000 recombinant males now that we know that our genotyping approach is going to work and is cost-effective, and then we need to develop some new statistical methods for assessing variation in the recombinational landscape, with the question of how much within population variation and between population variation there is. And now my ultimate goal is to identify cis modifiers of crossover distribution in Drosophila, and we can ask the question whether those cis modifiers mediate within population variation in the same way that they mediate between population or between species divergence. Right, this is a motivational speech, and so I've got to give you some lessons that we learned along the way. I mean, there's some scientific lessons that we learned, and those are kind of specific to this experiment. But I think the bigger lessons to be learned, science isn't pretty, right? So we read beautiful papers, and we go to hear beautiful talks, but for every beautiful talk that you hear, there are dozens of lab notebooks in the lab behind that document experiments that maybe didn't go as well as planned. And a related lesson is that sometimes less than successful experiments are really valuable, and you can learn a lot from them, not only to design more powerful experiments, but also sometimes the reason experiments don't go well is because of interesting biology, and there's something to be learned there too. Finally, we have definitely learned this lesson the very hard way, which is never underestimate the power of a pilot experiment. So you can educate yourself by reading papers, you can simulate, you can calculate until you're blue in the face, and nothing compares to actually getting in the lab and doing something at a small scale to see how it's going to turn out. And no motivational speech is complete without a quote, so I will leave you with the immortal words of Benjamin Franklin, who said that energy and persistence conquer all things. And I'm convinced that we have finally conquered this region, and that this underlying question of fine-scale variation and recombination rate in melanogaster, we're going to learn something very exciting in the next year. I need to acknowledge all of my collaborators from the past 5 years on this question. Andy Clark and Chip Quadro were my postdoctoral advisors. This is Andy, this is Chip. They were in the fly room with me every day, scoring the white anakinase phenotypes. Eric Stone, I now call him Blood from a Stone because he really did manage to extract a believable and reproducible statistical signal from those garnet scalloped pooled resequencing data. Stephanie Ruja is a technician in the lab, and she was instrumental in helping do the second round of crosses with the 16 different lines. And then Peter Andolfato, of course, for his help with the multiplex shotgun genotyping. And so I'll thank you for your attention, and I'll be happy to take questions at this time.